Welcome to NewYorkBIO's virtual breakfast series, a digital program started in 2020, bringing you fireside chats with leaders from across the healthcare spectrum. This week's episode features Elizabeth Schwarzbach, the Chief Business Officer of the New York Stem Cell Foundation's Research Institute.
All right, good morning, everyone. My name is Jennifer Hawksbland, and I'm the CEO at NewYorkBIO. Thank you so much for joining us for another edition of our virtual breakfast series. And thank you to our sponsors, TriNet. We're thrilled to have them on board bringing the type of content that we've had. As we enter into the middle half, it seems like, of October, things keep changing. We're watching the news, obviously, lots being said around the coronavirus vaccine development. And we'll continue to watch that, the moves the FDA makes as well as the manufacturers. A lot of NewYorkBIO member companies are working in this space, and we're thrilled to be able to support the work that they're doing. But today, first, before that, we have an illustrious guest in Liz Schwarzbach from the New York Stem Cell Foundation. And I'm going to kick it over to Derek to introduce her. And a matter of housekeeping, which I forget like half the time. Below your screen, you have the Q&A and the chat feature. So please send your questions and we'll get to them in due course during the conversation. All right, thanks. Over to Derek.
All right, Liz, good morning, and thank you so much for joining us today. So I'm excited to talk about the Stem Cell Foundation because I almost think that for a long time, it's been kind of a hidden gem within New York City's scientific community. So for those of you that don't know, the Stem Cell Foundation was founded in 2005. For a long time, it was up at the Audubon Research Center up in Columbia. Mm-hmm. And now is in this gleaming new fantastic building in the Hudson Research Center on 54th Street, if I am not mistaken. So Liz, good morning. It's awesome to have you here. Do you want to give us a little bit of an overview of the Stem Cell Foundation and what it does?
Yeah, I would love to. So thanks so much for having me. It's great to be part of the NewYorkBIO community and to be here this morning. So I really appreciate the opportunity. So the New York Stem Cell Foundation Research Institute, or NYSIF, as we call it, is a nonprofit organization located in Manhattan. So as Derek said, it's on 54th Street and 11th Avenue. We are in a building that was actually rebranded after NYSIF moved into it, known as the Hudson Research Center. There are floors available that can be built out for lab space. And I would have to say, having been there now for almost 3 years, it is a fantastic place to do science. So a little plug for the building. Our mission at NYSIF is to accelerate treatments and cures using stem cell research for the major disease of our time. As Derek mentioned, NYSIF was founded over 15 years ago by our CEO, Susan Solomon. And that was really at a time when stem cell research was primarily embryonic stem cell research. This was an area that was under siege by our federal government. Therefore, there's a lack of NIH funding or any funding for that matter. So really, and I should mention, this is an area, again, under siege by our current administration. So something to continue to focus on. So there's a real need to build the pipeline of scientists and ensure that we didn't lose a generation of stem cell researchers. So initially, NYSIF had funding programs, and they still exist. Our external programs is run by Kristen Smith-Duty, who's part of my team. It's fantastic. It's really amazing what she's done leading this program. We have a NYSIF Robertson investigator programs. It's a $1.5 million grant over 5 years. And the RFA actually opens at our conference in October, which I'll talk to more. So if you are an academic lab, in an academic lab listening, please apply. And then we also have NYSIF Draca Miller Fellowship awards that are given to people in the tri-state area or alumni of our investigator programs in those labs.
And a lot of your funding supports earlier stage scientists and postdocs, right? So you're really kind of bringing out the next generation of folks that you hope will establish these really solid research bases with what you're doing. So it's almost kind of like a big Kickstarter or an accelerant on someone's academic career in the field.
Yeah, so the investigator awards are for early career scientists. So this was really to make sure that there wasn't, to encourage people to go into this field. And if there wasn't the opportunity to get an NIH grant within the first year or two of starting your lab, this was, that resource, and allows researchers really to do the high risk work that might not be funded by the NIH, and really be creative in the research that they're doing. And then the NYSEF Druckenmiller Fellowship is for postdoctoral fellows. So definitely was, that was the remit, was encourage people to go into this area or stay in it. And then, ultimately there was this need for a safe haven laboratory to actually do this work. And so that was the genesis of the lab up at Columbia in the Audubon building, was to allow for this, research to be done. So NYSEF has always focused on high risk, high reward research. We were the first to create a personalized embryonic stem cell, which was the time #1 breakthrough in 2011. In 2012, we really became the pioneers in mitochondrial replacement therapy, and having cures for these really rare diseases. Again, a very controversial space, and an area that NYSEF has continued to be involved with. And then funding and pioneering a lot of the work to show that you could use stem cells from patients to create a disease model. So it might be worthwhile just explaining. I mean, I think a lot of people on these calls may be from different backgrounds.
Yeah.
And so now most of the work, in 2007, there was the advent by Shinya Yamanaka, who actually visited the NYSEF labs last year after our conference, to be able to take a tiny apple seed size of skin and reprogram that, or set that back to the embryonic stage, so to say, thereby inducing a pluripotent stem cell. So this now is a cell that has the ability to change into any cell type in your body. You can change the stem cell into each of the different types of neurons. You can change it into the heart cells, your liver cells, etc. So if you take that skin punch biopsy, this tiny piece of skin from someone with Alzheimer's, let's say, or Parkinson's or ALS, what research that NYSEF has supported or done within our lab has shown is that you can create a way to study Alzheimer's in a dish, or ALS in a dish. So you have, a representative human model of disease, and that's what's really been valuable and opened the door.
Yeah. It's kind of like the precursor to all of the organoid research that's happened, right?
Exactly.
So,
yeah, and when you put them together, you make these 3D structures, and they have been really valuable, representative models, again, for brain disease with your brain organoids, or liver disease with your liver organoids. Yeah.
I think what people don't necessarily appreciate is that for the longest time, science has basically been driven by animal models and approximations of what we think a disease looks like. And one of the biggest things, in my opinion, of what stem cells have brought is the ability to really use human tissues to study human tissues, something at least closer to what we hope is going on in the patient, right? It's those steps towards understanding human biology and disease that are actually really, really important and occur 15 years before anybody's able to take a drug. And it's incredibly important, and you guys have been doing work on the cutting edge of that for ages.
Yeah.
So you also have an element of science education in what you do, correct?
Yes. So we do have a educational program. So we have both a summer internship program for primarily college kids, but also high school students. And we also have a running seminar of outreach. And then I should mention there's about 100 people now who sit at NYSEF, who are NYSEF employees, and they make up the NYSEF Research Institute that you can see over my shoulder. And around 70 of those are based in the laboratory. And they are software and hardware engineers. So they have built an automated platform, proprietary series of robots called the NYSEF Global Stem Cell Array. And this has allowed us to derive from many patients at one time stem cells from either skin or blood, and then differentiate them to different cell types I was mentioning, or gene edit them. So it's a fully automated platform. We also have biologists and disease area experts that then use those human models of disease that we were talking about and do understanding diseases, discovering new medicines. So really bringing that to life, so to say. And then most recently, which I think is really exciting for the New York community as well, is we have completed construction of our good manufacturing practice suite, so our GMP suite, which is now a lab that can make clinical grade stem cells. So everything you see in front of NICEF is to make research grade cells for the use of disease models. But transitioning that, one of the other valuable things about stem cells, if there's a disease where one cell type's dysfunctional or a primary driver of the underlying cause of the disease is a cell type, that cell type can be replaced. And so this is, a cell replacement therapy. So the GMP team has been built out over the last, few years. We now have our GMP suite, and we're really well positioned to bring our first cell replacement therapy to patients, which is for age-related macular degeneration. So it's really an exciting time, I would say, to be at NICEF and, within the New York biotech community more broadly.
Yeah. So just to add, so the Research Institute also serves as kind of a discovery engine as well. You do your own research, you are developing your own candidates and looking to try to get them to patients as well. So as a question and also something to highlight, so moving into the new space, I imagine, allowed you to expand a bunch of these capabilities. I don't know that all of that would have fit under the previous footprint, and that's being generous to the previous footprint. So I imagine this allowed you to, really kind of step out and explore being able to do things from the GMP standpoint.
Yeah.
That's fantastic. Go ahead.
And we have just also, extended the space that NICEF is in. So we now have two full floors in the Hudson Research Center. We started off with 1.5 floors. And so it's really exciting that we're building out this additional space, just given the need, based on the amount of research that we're doing.
Sure. So we'll get into kind of external relationships and biz dev a little bit later, but are you able to act as also, I don't want to say kind of a CRO, but are other people able to access your GMP suite? Is it one of the ways that you work with external organizations?
Yeah, so NICEF collaborates with, and we'll talk about later, but organizations across the life sciences ecosystem and is a valuable resource for, other companies within New York and beyond globally too. Yeah.
So Liz, when you talk about commercializing or getting to the point where you're ready to commercialize a therapy, how do you partner, or will NICEF follow that through all the way?
Yeah.
You don't seem to be set up for commercial distribution. So
yeah, no, we're not a commercial entity, but, I think that NICEF has generated a lot of IP that is now at the point where it's been sufficiently de-risked to spin out into for-profit innovative companies. So I do expect, it's, like I said, a very exciting time for NICEF. And I think these spin-out companies will further build the New York City biotech community. We also do partner with, pharma or other, we've spun IP out into other biotechs. So NewStem, which is a company that has IP also from Hebrew University.
Yeah.
So we, all of the above. Yeah. So, really as I mentioned, our mission is to, really impact the major disease of our time. So the path that creates value for NICEF so that we can continue the research and bring that, medicine to patients is really the path that we'll take for each of the different programs.
Yeah.
Yeah.
We had a good question in the chat asking, why is it a proprietary platform, and thinking a little bit about the missions of nonprofits to kind of diffuse, the information and innovations that they generate. So I'll let you run with that one for a little bit. I can imagine a few reasons why, but.
Yeah. So I would say, first of all, when, the ability to make a stem cell from skin or blood was first, invented, and the value of this technology, so that you didn't have the limited resource of embryonic tissue, really, and the ability to take it from patients. Really, the innovation that Susan Solomon, who is our CEO, and NICEF saw was, to be able to do population level studies, you need stem cells from many people, and in what this pandemic has shown too is just the healthcare disparities. We need it across a diverse population of people. You need to include, you know, all genders, all races in the research that we're doing. And so doing this by hand, you know, isn't as feasible, and being able to do it robustly, reproducibly, have, you know, a postdoc come in every day and make the same exact stem cell lines really challenging. So we would have bought a platform if that platform existed, but it didn't. So NICEF built it, and hence the reason it's proprietary. So they, you know, built, you know, methods that didn't exist elsewhere. And, you know, we do make stem cells we leverage this then platform and make stem cells for researchers globally. We've been, you know, had a long-standing collaboration pretty much since the inception of NICEF with Mount Sinai. We've made collections for them for patient populations like schizophrenia, where the genetics are so diverse, you can't study one or two stem cell lines. You need lines across patient types. So we've definitely, you know, feel that we've leveraged it for, you know, for the benefit of the community. We also do have a repository of these stem cell lines, which we make available to researchers, the academic researchers. It's really just the cost of us to distribute the line to them. You know, they are not, you know, paying for the creation of the line. So we do look to make this resource available for sure, and definitely agree with you that it's, you know, extremely valuable. I think for the part about the sustainability of the mission of NICEF, I think that there is value in research institutes creating intellectual property, because this is how you ensure it gets to patients. You know, a pharma company, a biotech company cannot have investment if something's not protected. So that is that need to protect it, to get it to the patients, and then ultimately have that value come back to NICEF.
Yeah, for sure.
There's also a bit of a time sink too, where if you're, you know, distribution isn't a free, no effort situation, right? If you distribute something, people are going to ask you how it works, and you're going to be you need an actual staff to support it. And, you know, especially if you have something that is, say, flexible or not exactly a push button system, you could spend your entire life explaining it to everybody, and trying the effort to get them up and running and using it is probably greater than just having them collaborate with you in the first place. Right. So not everything that's a trade secret, or not everything that is proprietary is an easy out of the box solution that you're just keeping in a back room.
Yeah, and I mean, as you mentioned, Derek and Jennifer, you know, it does take funding to, you know, have the scientists and the engineers that I've discussed, and even the programmatic staff to do the educational support across New York City. So given the urgency of this work and the high risk nature, NICEF does rely on philanthropic funding for its research institute. So to that end, I would give one plug. We do have our virtual gala coming up. So this is, oh good, did you ever? Yeah, it's Show Up for Science this year. I think Christine Lynn Egley, who's our Chief Advancement Officer, came up with that, and I think it couldn't be more apropos for our time. And so it's being held October 27th. I'm gonna put the information link in the chat, but I would encourage, you know, the one benefit of these virtual events is that it really takes down the barriers for attending. So if you want to learn more about our specific research programs, digging into the science that we're doing, I think it will be a really informative night and fun. Hopefully it's going to be 1 hour, 7 to 8. It's being directed by Scott Ellis. So we've been really fortunate to have a Broadway director directing it. Whoopi Goldberg is going to be featured in it. Jesse Tyler Ferguson's interviewing our cancer researcher, Billy Porter. So I think it's, you know, going to be a really important night, and raise the critical funds for NICEF. So
yeah, it's I looked at I mean, I want to listen in just to learn, but I looked at the lineup and I was like, you know, I can easily devote an hour to that, because what a great lineup of folks you have. And I feel like that there are going to be some super cool videos of the work you're doing that we can share with the community even afterwards if they can't attend on the 27th.
Absolutely. Yeah, and then we also do have funding from grants and research collaborations, and New York City and the New York State have also been, you know, important partners as we built out these labs. So yeah, so I would say takes a village, as they say, and it's definitely
Yeah.
Whole city, a state.
Yeah. Well, I'll tell you that, you guys moving into that building, I think it's now, I think we've found with each new research center that goes up within the city, I think it impacts the community in this really great stepwise fashion, right? And I think more people tend to take notice of what's there. More people, it drives traffic one way or another to get there. I think people can talk about where things are in the city in kind of disparate locations, but it doesn't seem to me to really where something is located within the city doesn't really seem to me to impact the fact that people will go there, and it's definitely drawn a lot of attention to you and your team, to the folks at Hybercell, and really kind of adds another destination for people to go within New York.
Absolutely. And I think it's, easy enough to get down to the New York Genome Center, which is a straight shot down, going across town, as long as it's not, the UN, there's certain times when you don't want to go across town, but going across town, it's easy enough to get to MSK or Mount Sinai, straight up to, Columbia. So I think, we're really well connected within the Institutes of New York. And that was really why locate in Manhattan.
I walked to your office from Grand Central too, so it's
Mm-hmm.
it's easy.
Yeah,
it's not too shabby. So if we're thinking about this, you have a big team within this brand new research institute, and March rolls around and COVID hits. So how have you guys been dealing with the pandemic? I know that other places have had really creative ways to try and deal with it. Other people have kind of shut down and gone completely virtual, but how have you guys handled it?
Yeah. So NYSIF has remained open and operating throughout the entire pandemic. So from about March 12th through May 18th, when we were really within New York, at least at the height of the pandemic, we had a core team of engineers who maintained all of the ongoing reprogramming, differentiation, quality control, and expansion runs. So this was really possible because NYSIF was designated by the NIH as essential. I think that was a big motivational boost when we received that letter of designation, given that we do have collaborations with the NEI, the National Eye Institute, with Francis Collins Lab, working on diabetes. So I think that was really helpful for our scientists to say, this is something we need to do. And it's a heroic effort that they were able to go in. It was also possible because we have this end-to-end robotic platform. So it does require, tip changes and, kind of oversight. But I think, this definitely wouldn't have been possible to keep all of that, running. I think in the time from March 12th through May, we were able to maintain and complete 600 iPSC lines across all of those processes that I described. So, that would never be possible. And I don't think, it could be done if you were doing everything, by hand. We maintained our collaborative projects. So, things may have slowed down, but, everything was, really kept on track. We did, to ensure the safety of those that were there, the core team, we did have all other NYSIF staff work remotely. And I have to say my team has been amazing. I have a BD team. Jeff McGrain's our Director of Business Development. Corvus Richardson's our Senior Director of Legal and IP. Krista Voltaire's our awesome coordinator. They've really been, as or more productive during this time. And then we did move a lot of our in-person activities. The first question that came up was we have these seminars planned. Right. What are we going to do? And Rachel Ayer, who oversees all of our scientific outreach, really moved that content into, a virtual platform. David McEwen, who's our CEO, and Hallie McKean, who's oversees events, built out a virtual platform to do everything from our retreat. We have an annual retreat typically held in Montauk each year.
Oh, yeah.
That was held virtually. And I have to say, there still was, it's not the same, and we all miss being there, but really was.
Virtual Montauk is not the same as regular Montauk.
We all had, the images of Montauk up in the background. But everything that we do.
I could have made an argument for beachside, low risk.
Social distancing. It's the travel. So we have innovators from around the world. So I did say maybe the people in New York should. No, we do have, innovators in Europe, Israel, Japan. But even those in Japan attended, so it was really amazing.
That's awesome.
Yeah, I'm glad that you keep everything going, because just for the edification of the audience, these are not, independent, discrete experiments, right? These are long-term experiments that end up in a cell line that has to be maintained. And, it's almost like gardening in a way where, you don't actually get to the end point unless you actually do all of the steps in between. So honestly, an interruption in one thing could be catastrophic. It wipes out, so much work that it really doesn't get exactly. So it's great that you were able to keep going. I'm really glad that the processes were able to keep going, because again, these cells in a lot of ways are the engine by which so many people conduct their research. Yeah. So you also have your annual conference coming up next week. There's another big conference, the meeting on the Mesa, that is a big conference for the stem cell industry. I'm interested in how kind of the whole stem cell community has either come together or maintained, what it's been trying to do throughout COVID, just given the actual physical labor intensiveness of most stem cell research.
Yeah. I mean, I think it's been amazing to see actually how the stem cell community has come together. And really this pandemic, I think, has highlighted the importance of biomedical research, the importance of, understanding disease in different populations of people. So I should mention, NYSIF has been doing, immediately thought, how can we do meaningful research to contribute to the COVID effort? And we started to optimize the way in which we can take a stem cell and differentiate it to a lung bud, or kind of this spherical structure, and then invert it so you actually can study those lung cells. And so we've been doing that and working with infectious disease labs then on on COVID. So we're making ACE2 expressing cells. And then, even prior to that, NYSIF was using our 3D printer for, our colleagues at Cornell to make the, face shields for their PPE for, so everyone was kind of like, how can we pitch in and do, the most for combating this pandemic, recognizing right away the extent to which it would affect, our health and our society. The New York Genome Center has had the biweekly consortium calls that have highlighted just the, immense work being done across New York within the hospital systems, the convalescent plasma studies that have been done from, almost like week one. So I think that's been great. ISSCR has had Thursday networking sessions where they've highlighted, stem cell research being done. So I think, there's really been this kind of this collective effort. The meeting on the Mesas is this week that you mentioned. That's one where from a nonprofit research organization, most of the sessions are more focused on, company pitches, et cetera. So I usually go there to meet with colleagues in the field. So I feel like, that this is the first year I'm not attending that because I think that, isn't as much recreated in the platform being used. But I think the NYSIF annual conference you mentioned is next week. I think that we have really put in play, we spent a lot of time thinking about what the production company and platform would be that would allow us to retain that personal connection. So allowing you to video chat with, any attendees, et cetera, having an interactive poster session, not having any recorded talks, et cetera. So I really think we have put in place a stellar agenda. Tony. Oh,
your agenda is great.
I'm really excited.
Tony Coles, Carl June.
We have, it's across therapeutic areas. So we have Helen Blau, Chuck Murray, Christine Mummery, and cardiovascular. You mentioned Carl in cancer, and Irv Weissman. Actually, Anthony Johnson, who spoke last week on NewYorkBIO, is going to be speaking. So it's really, really amazing. The list goes on. So I'm also going to put the link to that in the chat to information.
We can share it with everybody too.
Yeah.
So encourage you to register. I would say, it's been a challenging year to secure funding. Things have been delayed, so as though we kept everything, maintained. It's definitely harder to garner sponsorship support this year. And, grants delayed. So it's, I think, you know, your support is really welcomed and appreciated.
Yeah, well, you know, I think everyone is in the same boat. And I think you, especially when, you know, we thought about this with our meeting too, you think about, you know, how are you going to reach people? You know, what is your content going to look like? How do you need to change? How do you need to be there for your audience? And in a lot of ways, I think it's made you be a little bit more creative, right? You know, I think people, I think, you know, there's the standard things that everyone can do, but I think everyone really has thought hard about how they actually connect with their audiences and what are the kind of things that their audiences need to see, what's going to be educational. I love the idea of poster sessions where you're on video chat the entire time. I think that's great. And, you know, we talk every week about how, you know, biopharma has put its best foot forward in terms of innovation and things during COVID, which it absolutely has. The other thing I think is really interesting is that I think people have shifted pretty seamlessly to the tech and communication tools. I think they've found, you know, new and good ways to use virtual platforms so that, you know, we now all have this different, you know, tool in our toolboxes that, you know, we can use to either communicate with other scientists, that we can use to communicate with businesses, with patients, with other stakeholders. And that's really interesting. So just for that alone, I'm going to register and go next week. I think that's really exciting. I forget whether you've overlapped with our breakfast series next week, but I'll forgive you if you have.
Well, we don't start until 9:30 on Tuesday.
Okay.
So you could do, you know, half and half.
and
Tony and Susan
nothing happens in the first half hour anyway, so everyone can attend the full breakfast series and then go on.
And then pop over.
Exactly.
Yes, and then we can pop over. So that's really good. I'm gonna encourage everybody to go and do that, just because, you know, you guys put on a great meeting every single year, and, you know, again, I think it highlights some of the best things in the entire stem cell industry. So we usually do this first, but we got talking about the foundation and things first. But I want to transition a little bit to your background and you know, talk a little bit more about you and where you came from, because you've actually spent, you know, a long time doing, you know, strategy and business development, literally on, you know, many different sides of the scientific industry, right? You've been in larger companies within biopharma. You've been at multiple not-for-profits. So do you want to give us just a little sense of where you came from and how you got to the Stem Cell Foundation? Yeah,
definitely. So yeah, I think if you look at my background, it may seem a little bit, I don't know, ADD, going between biopharma and nonprofit and consulting, which in and of itself is like for people that just like always to do different things. But I think, you know, one thing, you know, I've always been passionate about drug discovery. So I think, you know, the mission is really what's driven my decisions of where I've, you know, kind of focused, et cetera. You know, I started off in the late 90s at Merck. At a time that was a really exciting period for pharma. I remember my first day there was the day Singular was approved, and they had this huge barbecue on the lawn. So I'm like, you know, like newly minted graduate of like, you know, out of undergrad, and I'm like, this is the best thing ever. Like, colored M&Ms at lunch, like, you know, it was like the heyday of pharma, and I thought like life couldn't be any better. And
were you at White House Station?
No, I was in Lansdale in Pennsylvania, at their large site there, which now is kind of many sites, but it was West Point was the site. And so I thought, you know, and I was a biologist. So I was at the bench, and I quickly learned that, you know, most drug programs failed. The 9 preclinical programs that I worked with, 1 made it to the clinic. So, you know, thinking about, you know, this is where I became very comfortable with attrition, learned what drove it, I guess, too. Went to realize that I didn't know, you know, as much about pharmacology, and went to do a PhD in neuropharmacology at Penn. And that was where, you know, I really fell in love further with kind of the discovery component of you know, finding new medicines. But it was, you know, a bit burnt out from animal research, which is, you know, ironic since at that time induced pluripotent stem cells hadn't been invented, where now you can work in vitro with and you know, kind of work in the lab without necessarily having that be a factor. But went on to do strategy consulting. And that's where I really, kind of experienced the different exciting parts of the ecosystem.
Yeah.
I think consulting builds such a valuable toolkit for the, business development area of, of biopharma, and went to GSK in the R&D strategy group, and loved my time at GSK. Worked with people like Moncef Slaoui, who's now on the Warp Speed. Raked across the coals, and I just,
oh god, yeah. Now he's getting killed for owning GlaxoSmithKline stock after he was the head of R&D for a decade or whatever.
Yeah, which was his retirement. Yeah, exactly.
Yeah.
And he said any profit that he made in his retirement during the time that he was on Warp Speed, he would donate to COVID.
Yeah, no, he can't win. It's really terrible. We've got somebody that, spent however long doing, basically the perfect candidate to run that kind of operation, who is it's too bad. Anyway, back to you.
Yeah. And I mean, that's something that NewYorkBIO is really, important. And the bio industry actually is so critical and important, is just informing the general public about the role of biopharma. I have so many friends who think that we don't want to cure disease because that wouldn't be profitable. And of course, that's anyone that, everyone on the boat today, I mean, anyone in this area, our only mission is to improve the lives of patients or people suffering.
You're right. We do a ton of work, both at the national level, at the federal level, right? With particularly in this case, with our New York representatives. And having been on the GSK team, the Federal Government Affairs team, that took Moncef to the Hill for a hearing on Avandia, I definitely know how that is. But we also work with the state, because if you think about it, it's, the discussion, and certainly now in our current political climate, the discussion is all about the federal politics, right?
Yeah.
But state politics are really important too, and policies about how access to medicines and reimbursement. And so, so we do a ton of work educating policymakers, but also the public, because you're right, now it's intertwined.
Yeah, and I mean, I think this pandemic too just highlighted also, just the fluency, the scientific fluency that our public has, and it's just so important, as scientists, as people within this industry, to have conversations with people. I think it's almost like a topic that's avoided, and I think it's just so important to make people comfortable, with the topic. So it's not something that they're scared of or think is a mandate or, view that way. They see it as an opportunity to, engage. I moved from biopharma into the mission-driven organization, not say biopharma is mission-driven, but into nonprofit organizations. Really, someone I worked closely with at GSK, Perry Neeson, who had headed up the oncology therapeutic area and then was the SVP of Science Innovation, became the CEO at Sanford Burnham Prebys out in San Diego. And that was what brought me across the country. I knew that I probably wanted to come back to the East Coast, and they were a basic science institute that had a really robust screening center, over 1 million compound library, and was looking how can we change from that basic science institute to a more translational institute. So went there to kind of build the strategy to allow them to do that. It's great to see them, succeeding. They have compounds now in the clinic, and have, some really meaningful partnerships that we have put in place when we were there with companies like Eli Lilly, Daiichi Sankyo, Takeda. Really, I think had worked on that transition, and then came back to New York actually to join Axovant, a biotech, that's part of the Roivant group. They were developing intepirdine, which was a phase 3 compound for Alzheimer's disease. It was from GSK, so it was at GSK when they out-licensed it to Axovant. I thought it was a unique opportunity because David Hung had just taken over as CEO, so they had a scientist at the helm. I had known Vivek Ramaswamy, the CEO of Roivant, from when I was in consulting. I had done a lot of work within New York when he was at QVT, the hedge fund he was at prior to Roivant. And so I thought this is a unique opportunity to head up BD. If interberdine is successful, which I kind of put a 2% chance, even though the street was interestingly predicting much higher. If it's successful, there would be a windfall of funding to create a really neat pipeline of, from academic. I figured I'd just come from Sanford Burnham, which had a really strong Alzheimer's program. Thinking about the academic space, there would be opportunities to in-license really early stage programs and across the neurodegenerative space. And if it failed, there would need to be a kind of quick win, something, a BD to pivot on. And I think when it did fail, it lost $2 billion of market cap. I think the first 6 minutes the market opened, and they, there's a tailspin at Axovant, and I could have moved into Roivant, the parent company, but that really wasn't what I was looking for. And I thought, this was a compound that was discovered in mouse models, as Derek alluded to earlier. Mice don't get Alzheimer's. They don't even have some of the populations of neurons where Alzheimer's is thought to start. And we really need to move to more human disease relevant models. And that was what brought me to NICEF, is NICEF's working on human drug discovery. And so that was really what drove me to go back into nonprofit when I kind of made the switch out. And I think NICEF is really uniquely positioned as a nonprofit. They have the ability to do this unfettered research, yet they don't have kind of the bureaucracy that you have, good, bad, or ugly, that you would have at larger academic centers. So, I think it is, a really unique institute and place to do science.
Yeah, I think in terms of innovation, I think the pendulum has swung very far back onto the side of academic science and academic discovery, and then being fueled for translation by either biopharmas or even through venture funding into biotech companies. So when you think about where you sit within that spectrum, can you talk a little bit about what kind of transactions or collaborations the Stem Cell Foundation tries to do in order to move its therapies forward?
Yeah, sure. So to start off, NICEF collaborates with partners who have complementary resources and aligned mission, across the ecosystem. So whether they're disease foundations, we've worked with many disease foundations to build really foundational resources for the disease research being done. We've partnered with other nonprofit or academic institutes. I mentioned all the work that we've done in Alzheimer's, PTSD, schizophrenia with Mount Sinai, but our macular degeneration program, our clinical partners, so the ophthalmologists or surgeons who actually be doing the implantations are at Columbia. We partner with Columbia on the clinical side of that. We partner with technology companies. So within, again, sticking with our New York partners, we have a partnership with Google that's been a long-standing collaboration to really accelerate integrating AI and machine learning into our drug discovery. And actually, we have a really neat pro bono program right now with 2 Sigma. So one of the unique things about being in New York is just the talent. I think, you might not think of New York as being a tech center. You think of Silicon Valley, but just the number of computer scientists and bioinformatics and data scientists that Google, Facebook, 2 Sigma employs. Susan and I were speaking with, one of the leaders at 2 Sigma who mentioned he had, I think it was 700 data scientists. And we were like, wait,
it's amazing.
Yeah, and as a nonprofit, it's really difficult then when you're competing with these other organizations for these data scientists.
Yep.
So, they have a program called Data Clinic. 2 Sigma has a program called Data Clinic where they partner with nonprofits. It's really motivating for these data scientists who are typically just working with financial data to focus on biological data. And, to them, it doesn't look different. It's numbers coming in, ones or zeros, So whether you're programming for, a financial trade or how stem cells grow. So we've been partnering with 2 Sigma on really optimizing, a lot of the databases, things that sit behind a lot of the automation. So it's really been fantastic to leverage the, broader New York ecosystem in that way and the talent that's here. That's going to become a much higher demand within the,
Right.
the bio arena,
Yeah.
Yeah.
Well,
Yeah.
data sets are just going to get bigger, right? And biology is a very complex thing.
Yeah.
And, I think one of the big things that, Flatiron Health kind of showed everybody is that, data curation and how clean your data is and what you do with your data before you even touch it with an AI engine is super, super important. So, the more as things get bigger, getting smarter about what we do with our data, how we take care of our data and where it goes is hugely important if you want it to mean anything. Because there's never a place where garbage in or garbage out was more true than with biological data.
Yeah. And again, this is where, automated processes really lend itself to something like machine learning and AI, because you need the data to be exactly the same every time.
Yep.
If an image is slightly different on, day 15 from one patient and day 15 from another, batch of that you can't do, you can't apply AI to that. So I think that's where, the volume and the quality of data really, is a unparalleled tool,
we had a good question a few minutes ago. Have you guys thought about ever setting up a philanthropic venture fund, kind of like what the JDRF has or what some of the other, the Cystic Fibrosis Foundation or anything like that? Have you ever looked at that?
So we've thought about it. We have, we've spoke to, both board members and those involved at the JDRF fund, the T1D fund. And so we have looked to that. I think there's so much within NYSIF right now. That's really the focus is, translating that. So we've kind of stayed focused on that. And I think this is the year when a lot of those things will come to fruition. But yeah, we have looked to see, does that make sense? Particularly funding. I would say there is, and I think this is, Derek, one of the questions we were discussing, there is no dearth of funding right now in the stems, in therapeutics for stem cells. So I think it's a really exciting time for when things get to the like venture funding level. You look at just the number of different companies that have received over $100 million to start stem cell companies. Blue Rock, located in New York.
Blue Rock, Century, all of it. It seems like every week there's another one that really just kind of raises the bar.
I think the T1D fund was set up because there wasn't that funding going into diabetes, whereas I think our thing is we have to get to that crossroad.
Right.
Yeah, and then on partnerships, we also have research collaboration agreements with biopharma, again to advance programs to patients. So one example with the New York Cytovia, where we're working with them. Collaborating on cancer programs, but
so have you seen, if you think about things through the lens of a not-for-profit, when if either an investor or a company comes and talks to you, is it different than if they were talking to another biopharma company? Have you found that the discussions that people have with you on the business side is different because you sit within a not-for-profit rather than just rather than if you were at GSK and you had a program that people were either looking to out-license or collaborate with?
Yeah, no, I don't think that diligence is different. I mean, I think that, they're depending on, the focus of an academic lab or the focus of a nonprofit or the level of funding that, they've had, the expertise, et cetera. I think the diligence is the same. People pay for what, they're going to get. So if there hasn't been, if the data package doesn't look the same from, if the same level of what would be done at a pharma and at a nonprofit differ, then you just pay less for that. I mean, I think that's one of the things that initially was discouraging when pharma started working with academic institutes was, wait, you're going to pay me $100,000 for a target, but I've been working on this target for 20 years, so
right.
I think, but if you have intellectual property and you have validation of how it's worked and you have done the right experiments right the first time and you've done that killer experiment, I think it's, have you done the killer experiment? If you haven't, what is that? How much is it going to take? And what will I get when I get there?
Right.
Yeah.
I think, in general, I think people within academic, we'll call it tech transfer or academic business development, have become much, much smarter about what moves the needle in terms of the value of an asset, right? For a long time, I think people within academia were interested in doing, the experiments that would get published. But now with the proliferation of tech transfer offices and people getting smarter about doing deals, there's a lot of even pieces within academia that have popped up to actually take the assets a bit further and really develop the value, still within the academic structure.
Yeah.
So it seems like the whole population has gotten smarter about what makes an asset more valuable.
Yeah,
which actually is better for patients, right? I mean, because you're more likely, the more de-risking you can do early, to be taking candidates into development in the clinic that might actually have a better chance of getting there.
Yeah, absolutely. Yeah, I think, 10 to 15 years ago, there was much more programs driven by aims 1, 2, and 3. And now programs driven by what are the fundamental experiments that I need within a data package to partner this and get it to a patient.
Yeah. It seems to me to basically, it raises the floor for, what is an asset that you can actually try and take out to the marketplace. Yeah. Right. And that means it can come from anywhere. Companies have to do the same thing. Academic outposts have to do the same things. But in general, when an asset is basically brought to somebody for out licensing or any sort of deal discussions, there's a set level of expectations regardless of where it came from.
Exactly.
And I think that just makes it better for everybody, right? It levels the playing field a bit because the types of things that are actually going to drive value in the marketplace. So. And I think that change has probably come about within the last, I would certainly say within the last decade. Right? I think things have really kind of risen across the board.
Yeah. And I think, the percentage of nonprofit researchers that had translational visions has just increased. I think that that really has been, on the increase. And I think at NICEF, we're working, as I mentioned, in this human disease relevant drug discovery. So, the tools that we're using, I think, really are amenable to, really robust partnerships. And then also the stem cell replacement therapies. This is kind of the next modality of treatments that we're going to, experience and, is going to proliferate, for treating patients or even functional cures for patients.
Liz, do you have a combination? Do companies come to you and say, we think this could be a candidate for researching via the stem cells that you all have created? Or do you go out and look for companies or entities doing work that you think matches what you all do?
Yeah, definitely both. So, we go to conferences, we prospect, our researchers are interested in, other research you're doing. So we definitely do continually survey the field and think about who are the target, partners that we would want to work with. And then also anyone can email partnering@nicef.org if you want to have a discussion around opportunities. But we also get, probably more inbound, either people looking to work with certain disease models that we've built out. So I was saying, if it's like brain organoids in the area of Alzheimer's, or, Valentina Fasati's done a lot of pioneering work in certain cell types that are now being shown to be extremely relevant in neurodegeneration, things like astrocytes, microglia, oligodendrocytes. So there's very few people that can make those cells. So they'll come, to us for that resource. So definitely our business development goes both ways. And like I said, I think the cliche of if 1 + 1 is much more than 2, that's who we're, interested in working with. It's cliche for a reason. So
we've had a couple of questions in the vein of what I'll call public communication, right? And, we've talked a lot about where stem cells are really important. And, there are other challenges to working in the stem cell field in terms of public perception. So I'm going to kind of amalgamate these questions into one, and just, how do you guys think about both the education of the public and, where you sit in terms of what people's perception of the Stem Cell Foundation are simply because of the field that you work in? Can you talk a little bit about that? Because I think that's important.
Yeah, so I mean, that's really, a lot of the early work that NICEF did was advocacy. So, the whole stem cell research field and the lack of funding was based on a misperception, that aborted fetuses were being used for embryonic stem cells, which, is not the case. Anyone who themselves have gone through in vitro fertilization or, have friends who have, there's a question that says, what would you like to do when you no longer want to maintain the embryos that you've created? And 96%, I think, are, a vast majority of people select donate to research. And that's really, actually a valuable way to use that tissue for stem cell research. And there's been this misperception from the very onset that's really led to, it being under siege. So, I mean, we understand the need for education and for untangling things that, misconstrued, get misconstrued from the beginning, from the onset. And so I do think it's really important. I mean, the one way that we focus on tackling this is, starting early. We have STEM tours for 4th through 9th graders, which is one of the key times when kids decide what career they want to pursue. So I think if, they say by 9th grade, you're almost too late. I don't know if that's true, but so, we really try to at least make it more accessible. A lot of kids, and, particularly now in the virtual world, you don't have a scientific curriculum, and it's almost, it's really difficult, even giving funding of schools, just how much is trying to be accomplished within a school day. They don't have access, so they don't know what they don't know. So we're really trying to have just even tours of NICEF for 4th to 9th graders. We have seminars on each of our disease areas. So every other month, they're free seminars. Anyone can attend. We've, recently had them on COVID, what the work that we're doing on COVID could do. So yeah, I do think it is just information. Information's king. And the problem right now with information is there's a very low bar to get information out. And so people have, disparate quality of information that is, informing their decisions. And so I think, we're trying to, as an organization and then as part of the broader, ecosystem, trying to really distill in a very easy to understand way what can be accomplished with this type of research, what can't be accomplished. So, I mean, equally important also in the stem cell world is, stem cell tourism and people using stem cells that are, adult stem cells, not the induced pluripotent stem cells that we've talked about that are differentiated to a specific cell type, but, your adult stem cells to cure anything. And, again, it's all about the studies that have been done to show that there is, proof behind the claims.
Right. You raise a good point too about, education. I mean, clearly, like if we did more education and people listened better, often we could, I think, shortcut a lot of the public debate, right, around things. It's a combination. But as far as children, I'd love to reach out to you as New York Bio works with the task force we're pulling together on equity, diversity, and inclusion to talk about your outreach to school children, as well as how we could partner on that. We particularly think about underserved communities and communities that have a low interest in going into science. I think that could be a key partnership that we could look forward to.
We had our first immersive high school program this summer. It was supposed to be in person. 3 day, I think it's 3 days or 4 days program. It was supposed to be in person, but we had it virtually, and it was amazing just to see the diversity of the students who participated and how interested they were once they started learning about what stem cells were and ways that they could be applied. And so I think it's a very fertile ground. And I should mention NICEF does have an initiative for women in science and engineering as well, the iWise program. We require every one of our applicants to any of our grant programs to fill out a scorecard that basically says, not only your classic, like what percentage of tenured professors are female, but also things like how many people on decision-making committees are women. And, most don't even track this information. So I think awareness is the first step. We recently published a paper, I guess about a year ago, showing the findings. And, over the last 5 years, there hasn't been significant change. So, I think it's an area that we continue to work on in addition to all the DEIB efforts.
Yeah. Well, it's a big area of focus for us as well. And, you know, just for our audience, we tend to follow these up with either a LinkedIn post or a Twitter thread. I'll include all of these links in what we put out today and this afternoon. Liz, we can't thank you enough.
Thank you.
This is fantastic.
Great.
You guys are such a big part of New York City, and you know we're super happy to support you in what you're doing. I'm excited for your conference next week and your gala in a couple weeks. But thank you so much for joining us this morning. We really appreciate it.
Thanks.
All right, have a great day.
Thanks, everybody.
All right. Bye-bye, everybody.
Thank you for tuning in to New York Bio's virtual breakfast series. Join us every Tuesday at 9 a.m. for more discussions with leaders from across the healthcare spectrum. For more information on New York Bio, please visit us at www.newyorkbio.org.